As we have discussed body systems, we have discussed the need to maintain homeostasis within each body system. Fluid, electrolyte, and acid-base balance require interactions of several body systems to maintain homeostasis. Body water content varies with age, body mass, gender, and relative body fat. Infants are at least 73% water. The elderly are more like 45% water. Young males are about 60% water, young females about 50% water. Adipose tissue is the tissue that will have the least water in it, containing only about 20% water. Muscle tissue will have the most water at 75%. Young males have more muscle tissue than young females, and this accounts for the difference in water values in both men and women. Water can be divided into two general compartments in the body. The intracellular fluid is all of the fluid that's inside the millions of cells that make up your body. The extracellular fluid is all of the fluid that's outside your cells. This will be plasma, interstitial fluid, and then there are smaller spaces that have fluid like the cerebrospinal fluid, your aqueous humor, your vitreous humor, synovial fluid, peritoneal fluid, things like that. If we look at our body as these compartments, and we look at our total body weight, about 40% of our body weight is in the fluid that's inside our cells, our intracellular fluid. 20% of our body weight is outside of the cells, basically this interstitial fluid and plasma. These are our extracellular fluids. Now remember, vitreous humor and cerebrospinal fluid are also part of this extracellular fluid, but the two major areas are the plasma and the interstitial fluid. When we are measuring electrolyte values or when we are measuring pH, we always measure extracellular fluid. It's very easy to get to the plasma. It's much harder to get to the interstitial fluid, which is all around our cells, and getting to our intracellular fluid is almost impossible. So most of our focus when we're talking about water balance and electrolyte balance has to do with our extracellular fluid space. Our body fluids are primarily composed of water. Water is the universal solvent. Then we have non-electrolytes and electrolytes. Non-electrolytes are those molecules that are covalently bonded. Covalently bonded molecules do not dissociate in solution. Typically, these are organic molecules, and organic molecules can move across the cell membrane by diffusion, facilitated diffusion, even active transport. So typically, these molecules maintain balance in the ICF and ECF simply because of their ability to move across the plasma membrane. The other thing about non-electrolytes is when we have one molecule of glucose in solution, it only accounts for one particle that's in that solution. So that non-electrolytes really do not contribute to the osmotic activity of fluids. They don't really draw water to them. Electrolytes are held together by ionic bonds. Ionic bonds will dissociate into charged particles. Because these charged particles will conduct electricity, this is why they're called electrolytes. Electrolytes don't diffuse. They don't move across cell membranes easily. Therefore, they're responsible for osmotic activity. If we have an imbalance of electrolytes, we move water to take care of that imbalance. We say that electrolytes contribute to the osmolality of a solution. And osmolality is simply a measure of how much pull there is for water from one side of the membrane to the other. When we say that these molecules dissociate, this means if we put one molecule of sodium chloride into a solution, we actually have two particles in the solution. That means that there's two particles to draw water instead of one. Electrolytes are typically measured in milliequivs per liter. ICF and ECF have to have the same osmolality, but the ions in the solutions do not have to be the same. If we look at our basic ECF solutions, plasma and interstitial fluid, you will see that the major cation is sodium, but there's very little sodium in the intracellular fluid. There's very little potassium in the extracellular fluid, but potassium is the major cation in the intracellular fluid. Calcium levels, magnesium levels, a little more in the intracellular fluid. Bicarbonate, not so big a difference. Chloride is the major anion in extracellular fluid, very little chloride in intracellular fluid. In terms of the anions in the intracellular fluid, we have phosphates and we have proteins. Water constantly moves between the ICF and the ECF. This is because of hydrostatic pressure and osmotic pressure. If there's any change in solute concentration in one fluid compartment, water will move to make this equal. Osmolality must remain equal in the two compartments. Exchanges between plasma and interstitial fluid, which are both extracellular fluids, occurs across the capillary walls. You'll remember this is driven by hydrostatic pressure and colloidal pressure, which is a special kind of osmotic pressure exerted by proteins. Exchanges between the interstitial fluid, which is part of the extracellular fluid compartment, and the cell, which is the intracellular fluid compartment, occurs as a result of osmosis, diffusion, and active transport across the cell membrane. We transport gases by diffusion into the blood, into the interstitial fluid, and into the tissue spaces. Nutrients have to come from the gastrointestinal tract, usually by active transport into the blood, perhaps by active transport or diffusion into the interstitial fluid, and then by active transport or diffusion into the cells. Water is constantly moving back and forth based on concentrations of the solutions in the ICF and the ECF. As ions move back and forth between the blood plasma and the interstitial fluid, water will also move back and forth. These ions can come out of our digestive system. Water is eliminated by the kidney. Nitrogenous wastes are eliminated by the kidney. We've discussed how this is osmosis and active transport based. The nitrogenous wastes have to get out of the cell. This is usually by diffusion into the blood. Ions move back and forth across the cell membrane, just like they move back and forth between the blood plasma and the interstitial fluid. As ions move, we're going to move water. To maintain water balance, the amount of water that comes into our body has to equal the amount of water that leaves our body. We have two sources of water. One is what we call metabolic water. This is water that's a byproduct of metabolism. This contributes a small amount of the water in our body. Most of our water intake is what we consider conscious intake. We drink fluids, we have water in foods that we eat. Water loss can also occur in two ways. We have the insensible water loss that we're unaware of. Water evaporates from our skin. We have moisture in our breath that evaporates from our lungs. Excreted water is the water that we're more aware of, sweat, feces, and urine. About 60% of our water loss is through our urine. Body fluid osmolality is tightly regulated between 280 and 300 milliosmoles. If there is an increase in the osmolality of the extracellular fluid, this will trigger thirst receptors and also trigger the release of antidiuretic hormone. If there's a decrease in osmolality in the extracellular fluid, thirst will be depressed and antidiuretic hormone will not be released. This will allow the dilute urine to leave the body. We take in about 2,500 milliliters of fluid a day. Metabolism water counts for a little bit of that. Most of it comes in food and beverages. That means that we have to put out 2,500 milliliters a day. Feces, sweat, insensible water loss, but most of it is in urine. Thirst is the driving force for water intake. Hypothalamic osmoreceptors will detect an increase in the ECF osmolality of as little as 1 to 2%. Dry mouth, a reduced saliva output, or a substantial decrease in blood volume, this has to be a 5 to 10% blood volume loss, will trigger the thirst center. So all three of these things trigger thirst. Water loss mechanisms include those obligatory water losses, the insensible water loss, the loss in feces due to the fiber in your diet, and it is necessary to make a minimum of 500 milliliters of urine to flush waste from the body. Additional water loss is dependent on fluid intake, how much water is in the diet that you take in, and water loss through other avenues. For example, if you sweat a lot, you may urinate less. Antidiuretic hormone will affect the collecting duct, causing water to be reabsorbed, conserving water for the body. One of the disorders of water balance is dehydration. This is when you have fluid loss of either water or water and solutes. Massive hemorrhage can be one way we lose water. Severe burns also contribute to water loss. Your skin is a waterproof barrier. We talk about it keeping stuff out, but it also keeps stuff in. If you lose a substantial amount of your skin, then you evaporate much more readily than you would think. Prolonged vomiting or diarrhea leads to water loss, profuse sweating, water deprivation, or diuretic abuse. All of these account for dehydration. In diabetes mellitus, because you're eliminating glucose in the urine, that causes more water to go out in the urine. And in diabetes insipidus, you don't make antidiuretic hormones, so you don't conserve water appropriately. The symptoms of dehydration include dry mouth, thirst, dry flushed skin, and a decrease in urine output. This is known as oliguria. If prolonged, there is weight loss, fever, and mental confusion. Because this can cause a decrease in blood volume, hypovolemic shock is a possible complication of severe dehydration. When there is excessive loss of water from the extracellular fluid, this is going to make the osmotic pressure in the extracellular fluid increase. As a result, to maintain balance between the extracellular and intracellular fluid, water will leave the cells by osmosis. This is an attempt to keep the osmolality equal between the extracellular space and the intracellular space. Another disorder of water balance is hypotonic hydration. Whenever the ECF osmolality goes down, it inhibits antidiuretic hormone release. This means that less water is reabsorbed and allows excess water to flush from the system as urine. In renal insufficiency, or if there is an extraordinary and rapid water intake, you can overhydrate. This is called hypotonic hydration. This means that the ECF is diluted. As the ECF is diluted, that dilutes the sodium, so you have hyponatremia, low sodium levels in the ECF. Hyponatremia causes osmosis of water into the cells. There's now more positive ions in the cells than outside the cells, and so the cells begin to swell. This leads to severe metabolic disturbances. The symptoms are nausea, vomiting, muscular cramping, and cerebral edema. The problem with cerebral edema is as the brain swells inside the skull, pressure is put on the brain. This leads to disorientation, convulsions, coma, and death. One of the places we've seen this is in marathon runners. They come in from that long run severely dehydrated. They take in mass quantities of water much too quickly. As a result, they have put in that rapid water intake, they've overhydrated suddenly, and because the body doesn't have time to do all of the adjusting it should do, they end up with cerebral edema. So in this case, excessive water enters the extracellular fluid, diluting it. The intracellular fluid is now too concentrated, so water moves from the ECF into the ICF to balance the ionic concentrations between the two fluid compartments. Edema is an atypical accumulation of fluid in the interstitial space. Here the imbalance is between the plasma and the interstitial fluid. Tissue swelling occurs, but there is no cell swelling. Edema typically impairs tissue function. It also increases the distance that materials must diffuse to get from the blood to the cells. The causes of edema are anything that increases fluid movement from the blood to the tissues, such as increased blood pressure or decreased oncotic pressure in the blood, or anything that inhibits the return of lymph to the blood, such as blocked lymphatics or too much protein in the interstitial space. Electrolyte balance usually refers to salt balance, but electrolytes are anything that dissociates, so salts, acids, bases, and some proteins technically are electrolytes. Electrolytes enter the body in foods and fluids. Some are even generated during metabolic processes. We lose electrolytes in perspiration, feces, urine, and vomit. People who experience an electrolyte imbalance may have a craving for salty or sour foods. This is very common in Addison's disease, where there's an insufficient amount of aldosterone. Some people even eat non-food items like chalk or clay. This is a condition known as pica. Sodium is the central molecule in fluid and electrolyte balance and in overall body homeostasis. Sodium salts account for 90 to 95% of the extracellular fluid solutes. That's why sodium is the most prevalent cation in the ECF. Normal plasma concentrations for sodium are 142 milliequivs per liter. You'll remember that the cell membrane is impermeable to sodium. Sodium can move between the ECF and body secretions, so sodium can be eliminated in gastric fluid and intestinal fluid, but it can't get into the cells. Also, as we've said before, water follows sodium. So when we change sodium levels in the ECF, this will cause changes in blood volume and also changes in the ICF fluid. There are no receptors for sodium. The sodium-water balance in the body is linked to blood pressure and blood volume, and the regulation of these is both neural and hormonal. Regulation of sodium is one of the most important functions of the kidney. Aldosterone is what controls the renal regulation of sodium. It also plays an important role in blood pressure regulation as part of the renin-angiotensin-aldosterone pathway. 65% of the sodium in the filtrate is reabsorbed normally in the proximal convoluted tubule, and another 25% is reclaimed in the nephron loop. That only leaves 10% of the sodium for us to do any real regulation of. If there is a high aldosterone value, the remaining sodium in the filtrate will be reclaimed in the distal convoluted tubule and the collecting duct. If there is a low aldosterone, then no further sodium is reabsorbed. The sodium is allowed to leave in the urine. Large losses of sodium in the urine lead to large losses of water in the urine. The most important trigger for aldosterone is the renin-angiotensin-aldosterone mechanism. This is triggered by low blood pressure and typically occurs as a result of triggering the juxtaglomerular complex in the kidney. Another trigger for aldosterone is high potassium levels. If there are high potassium levels in the ECF, this can directly stimulate the adrenal cortex to release aldosterone. The reason high potassium levels would trigger aldosterone release is because aldosterone will cause us to save sodium out of the filtrate. If we save sodium, we have to get rid of some other cation, and the cation we get rid of is potassium. So by saving sodium, we eliminate potassium. Low blood volume and low blood pressure triggers renin release. The sympathetic nervous system responds, not only causing vasoconstriction, but also stimulating the juxtaglomerular cells to release renin. Because there's a decrease in filtrate concentration of sodium chloride, then the macula densa cells also trigger the juxtaglomerular cells to release renin. And the decrease of stretch in the afferent arterioles trigger the juxtaglomerular cells to release renin. Renin will stimulate angiotensinogen to be formed into angiotensin 1. Angiotensin 1 will be converted to angiotensin 2, and angiotensin 2 is a potent vasoconstrictor, but it also stimulates the release of aldosterone from the adrenal gland. In Addison's disease, which is hypoaldosteronism, these individuals lose sodium and water. They stay on the verge of hypovolemia. They have a very hard time keeping their blood pressure up. Other hormones can influence sodium. One is atrial natriuretic peptide. This hormone reduces blood pressure and blood volume. Basically, it inhibits all the events that promote vasoconstriction and sodium and water retention. This hormone is released in response to stretch of the atrial walls. High blood pressure will cause too much stretch and will cause the release of ANP. ANP acts as a diuretic and it promotes sodium excretion. It also suppresses the release of antidiuretic hormone, renin, and aldosterone. Estrogens can act similarly to aldosterone, causing some water retention. Progesterone is a mild diuretic, causing some loss of fluid. Estrogen and progesterone are balanced during the menstrual cycle. Estrogen at the beginning of the cycle is relatively high. Progesterone peaks toward the end of the cycle. Most women experience some water retention and then lose that water as the result of the estrogen-progesterone fluctuations. When there are high levels of glucocorticoids, this can promote edema. High levels of glucocorticoids can act a lot like aldosterone. The neural controls for sodium include the cardiovascular baroreceptors. These very indirectly monitor sodium. Sodium controls blood volume, and blood volume determines blood pressure. When there is an increasing blood pressure, this activates the baroreceptors in the heart, the carotids, and the thoracic blood vessels. This causes a reduction of sympathetic impulses to the kidney. This means that the afferent arterioles will dilate. Filtrate production will then increase as a result of more blood flow into the glomerulus. As filtrate increases and more fluid is lost from the body, we will notice a drop in blood pressure. This will lead to reflexive constriction of the systemic arterioles, including the afferent arteriole. Filtrate production will be reduced and water loss will decrease. Potassium is the chief cation in the intracellular fluid. The plasma value is relatively low, 3.5 to 5.0 milliequivs per liter. Potassium levels are very important in maintaining membrane resting potential. The function of nervous and muscle tissue depends upon resting membrane potential staying relatively constant. The heart is particularly sensitive to potassium levels. Potassium is controlled by the kidneys. It's regulated in the distal convoluted tubules and the collecting ducts. Basically, it's either secreted or not secreted depending on levels of potassium in the extracellular fluid. The main thrust of the kidney is to excrete potassium, so you can become potassium deficient. Aldosterone will cause us to save sodium, and if we're saving sodium, we're losing potassium. So high levels of aldosterone can also cause us to lose large amounts of potassium. Some people have reduced their sodium intake by not using sodium chloride salt, but rather using a potassium salt as a substitute. This is a perfectly safe substitution as long as aldosterone release is normal. However, if there is a lack of aldosterone, hyperkalemia can result, and this can be swiftly lethal. This will cause hypopolarization. This means that the membrane potential will drop dramatically, nerve and muscle cells will not respond as they should, and typically what kills first is a heart attack. Adrenocortical tumors can cause the adrenal gland to pump out huge amounts of aldosterone. This can lead to hypokalemia or too little potassium in the blood. As a result, resting membrane potential goes high and neurons hyperpolarize. This leads to paralysis of muscles. One group of muscles that can be paralyzed are the muscles of the larynx. They can constrict and block off the airway, causing suffocation. 99% of the calcium is located in our bones as calcium phosphate salts. The extracellular fluid calcium levels are regulated by parathyroid hormone. Normally, we want about 9 to 11 milligrams per deciliter of calcium in the ECF. Parathyroid hormone will activate the osteoclasts of the bone, breaking down bone to release calcium to the ECF, and it will also increase calcium reabsorption in the distal convoluted tubule. When we increase calcium reabsorption, we decrease phosphate reabsorption. And finally, parathyroid hormone enhances intestinal absorption. It does this by stimulating the kidney to transform vitamin D to its active form. Vitamin D is necessary to absorb calcium. Most calcium is passively reabsorbed in the proximal convoluted tubule, and 75% of the phosphate is reabsorbed in the proximal convoluted tubule. Calcium is important for normal blood clotting, cell membrane permeability, secretory activity, and maintaining threshold values for excitable tissues. Hypercalcemia, or elevated calcium, lowers the threshold. This means that nerves and muscles are easily excited. This can lead to muscle tetany. Hypocalcemia raises the threshold. This means that nerve and muscles do not respond as they should, and this typically results in cardiac arrhythmias. Chloride is the major anion of the extracellular fluid. About 99% of our chloride is reabsorbed in the proximal convoluted tubule. Like water, chloride follows sodium. Anytime we're saving sodium, chloride is going to be co-transported with it. The other anions are the sulfates and the nitrates. They're typically reabsorbed in the proximal convoluted tubule to whatever transport maximums apply for those ions. Before we start talking about acid-base balance, let's have a quick chemistry review. You'll remember that acids are proton donors and protons are hydrogen ions. Bases are proton acceptors. They can pick up hydrogen ions. Acids and bases, like salts, are ionic substances, so they dissociate in solution. Things that are strong acids dissociate completely. That is, they totally unglue. All of their hydrogens go into solution and all of their anions go in solution. But weak acids dissociate incompletely. At any given time, only a few of the acid molecules are dissociated, so they contribute fewer hydrogen ions to the solution. pH is a measure of the hydrogen ions in solution. A low pH means it's acidic, and a high pH means it's basic. Buffers are special chemicals that can either absorb hydrogen ions or give them off. As a result, they help solutions resist pH changes. So here you see a strong acid, hydrochloric acid. Every single molecule dissociates, so we have the maximum amount of hydrogen ions in this solution. This would have a low pH. Carbonic acid, on the other hand, has some molecules that stay together, they don't dissociate, and only at any given time are a few of the molecules dissociated. So we have very few hydrogen ions in solution compared to the total number of acid molecules. Normal blood pH is 7.35 to 7.45. It's necessary that we maintain this very closely for normal enzyme function. Typically, if blood pH goes below 6.8, we're dead, and if it goes above 7.8, we're dead. Anything below 7.35 is considered acidosis of the blood, and anything above 7.45 is alkalosis of the blood. There are several sources of acidity in the body. Many of them are metabolic. For example, the breakdown of phosphorus-containing proteins will yield phosphoric acid. Fat, when it's metabolized, we get ketones, and ketones are acidic. And the anaerobic breakdown of glucose in the muscles yields lactic acid. All of these are metabolic sources of acids. Because carbon dioxide is carried in the blood as bicarbonate ions, we make some carbonic acid, which means some hydrogen ions are liberated as a result of carbon dioxide transport. Hydrogen ions can be regulated in three ways: chemical buffers, the regulation of the respiratory system by the brainstem respiratory centers, and by the renal mechanisms, those that help us eliminate bicarbonate ions. Let's look first at the chemical buffers. The most important chemical buffer of the extracellular fluid is the bicarbonate buffer system. Now remember, we can monitor the extracellular fluid, so this is the one we pay the most attention to. Carbonic acid is the basis for this buffer system, and as we just saw, it's a weak acid. You may remember when we talked about carbon dioxide transport, we talked about how carbon dioxide plus water, in the presence of the appropriate carbonic anhydrase, will yield carbonic acid. Carbonic acid dissociates weakly into a few hydrogen ions and a few bicarbonate ions. These bicarbonate ions were how we transported carbon dioxide in the blood. These are totally reversible equations, so they go back and forth as we need them to, depending upon whether we have a lot of CO2, which will drive it this way, or a lot of bicarbonate, which will drive it back this way. Carbon dioxide can be added to this buffer system via our respiratory system. So one way we can change pH is by monitoring carbon dioxide levels. Bicarbonate levels are controlled by the kidneys. So one way we can control this system is by regulating bicarbonate ions using the kidneys. This is the primary buffer system of the ECF, and it's easy to regulate because we can regulate it either through the lungs or the kidneys. The phosphate buffer system is a similar system with a weak acid that dissociates into just a few hydrogen ions. This is a very effective buffer system in the urine and also in the intracellular fluid. And the protein buffer system is based on proteins that are found in the plasma and the cells. You'll remember that one of the major anions in the intracellular fluid was proteins. We talked about a protein buffer system in the respiratory system. Hemoglobin, remember, can pick up some hydrogen ions and help buffer the red cell contents as we're exchanging carbon dioxide and oxygen back and forth. The respiratory system and the renal system help form the physiological buffering systems of the carbonic acid system. You need to remember that our partial pressure of carbon dioxide in the blood should be between 35 and 45 millimeters of mercury. When blood leaves the lungs, the PCO2 is at 35. When blood leaves the tissues, the PCO2 is at 45. So this is the normal level of PCO2 in the blood. If blood carbon dioxide levels increase, blood pH will decrease. Increasing carbon dioxide levels will develop more carbonic acid, which will release more hydrogen ions into the blood. The respiratory system then can be used to adjust pH by changing the PCO2 of the blood. If you increase PCO2 by conserving carbon dioxide, breathing more shallowly, you will decrease pH. If you decrease blood carbon dioxide levels by breathing more heavily, blowing off more carbon dioxide, then blood pH will increase. The other level we need to know about is the bicarbonate level. Normally, your bicarbonate level in your blood should be between 22 and 26 milliequivalents per liter. This is controlled by the kidneys. They actively excrete bicarbonate. They can even make bicarbonate if necessary and put it back into the blood. If bicarbonate levels decrease, so does blood pH. So if we excrete more bicarbonate, we're going to lower pH. If we save back more bicarbonate, we're going to increase blood pH. It is the kidneys that adjust for those metabolic acids, the phosphoric, the lactic, and the ketones, those acids that are made as a result of metabolism. Certain respiratory conditions can alter blood pH. In respiratory acidosis, we have a blood pH below 7.35, that's acidosis, and a PCO2 level that's above 45 millimeters of mercury. We see this in individuals who can't remove carbon dioxide from their lungs. These individuals are experiencing shallow breathing. They may have pneumonia or cystic fibrosis where less of their lung tissue is available for exchange of carbon dioxide and oxygen. In respiratory alkalosis, we have a blood pH above 7.45 and a PCO2 that's below 35 millimeters of mercury. These individuals are experiencing hyperventilation. They're blowing off too much carbon dioxide. Now, since our blood pH is out of range, we want to bring it back in range. We call this compensation. And if we have a respiratory problem, we're going to compensate by the kidneys doing something with bicarbonate. In respiratory acidosis, blood pH is too low. We want to bring it back up, so we'll retain bicarbonate because increasing bicarbonate increases blood pH. In respiratory alkalosis, the pH is too high. We want to bring it down, so the kidneys will secrete bicarbonate. This will bring down the pH and we'll get back to normal. There can also be metabolic imbalances that alter blood pH. In metabolic acidosis, we have a pH below 7.35 and a bicarbonate that's below 22 milliequivalents per liter. This can be caused by ingesting too much alcohol or by excessive loss of bicarbonate, as is seen with excessive diarrhea. In metabolic alkalosis, we have a pH above 7.45 and a bicarbonate level that's above 26 milliequivalents per liter. With excessive vomiting, you're losing stomach acid. That leads to alkalosis. Also, people who abuse antacids, that is, have excessive base intake, can develop metabolic alkalosis. Because this is a metabolic problem, we need to change the pH of the blood, and this time we're going to compensate with the lungs. We're going to adjust carbon dioxide levels in the blood to try to get the pH back to normal. In metabolic acidosis, we want to bring the pH back up. To bring the pH back up, we have to decrease carbon dioxide levels. So these people will begin to hyperventilate. In metabolic alkalosis, the pH is too high. We want to bring it back down. In order to do that, we have to increase carbon dioxide levels. So these people will experience hypoventilation. So let's see how this works. See if we can diagnose a patient. You have a patient that has a blood pH of 7.51, a PCO2 of 40 millimeters of mercury, and a bicarbonate of 28 milliequivalents per liter. It's important that you know the normal values for pH, PCO2, and bicarbonate in order to be able to solve this kind of problem. So the first thing we have to do is, what is the problem? Well, let's look at the pH first, because that's the easiest thing to do. His pH is 7.51. That's above 7.45. It's high, so he has some sort of alkalosis. How do we determine what kind of alkalosis? Well, we look at the PCO2 and the bicarbonate levels. His PCO2 is normal, so he doesn't have a problem with his respiratory system, but his bicarbonate level is low. This means he has a metabolic problem because bicarbonate tells you it's metabolic. So his condition is metabolic alkalosis. The compensation for this, the way we're going to fix this, is by using the lungs. In this case, we need to decrease the pH, and to do that, we have to increase the carbon dioxide level. So this individual will hypoventilate.